Okay, so we are going to start with development. So again, for the last 20 minutes, we have discussed the exam papers, but now we are going to move on to the lecture. So in development, what we are going to be learning is basically a continuation of last week's lecture, where we learned about the process of fertilization, about the process of ovulation, and about sperm generation. All of these we have already covered. So today, once the egg has been fertilized with the sperm, what happens after that? And we are going to be focusing on the human system. This will be a short lecture, but we are going to look at the basics of what happens to the egg after it has been fertilized with the sperm. So embryonic development begins with the fertilization or conception, as it is called. To conceive is to fertilize, which is basically the union of the egg and the sperm to form a zygote. So the shape of the sperm is highly related to its function. What is the function of the sperm is once it is in the female reproductive system, it will swim through the uterus all the way to the oviduct. So if we don't have a picture of it, but basically what it does, if you recall the image that I shared last week with you, just a moment, let me connect to my pen. Okay. So what we had learned last week is that there is a rather long tube called oviduct, which has this sort of, I'm not drawing it very well, but so this is where the uterus is of the female reproductive tract. So the sperm basically comes all the way to the uterus and swims all the way to the oviduct. Okay, so it goes all the way here. The ovaries are over here, and the ovary has ovulated, released an egg, and that egg has gone into the oviduct as well. Right? So the union of the egg and the sperm happens in the oviduct. This means that the sperm has to swim all the way from the vagina to the uterus and then into the oviduct before it can be fertilized. So the shape of the sperm is highly related to its function, which means that the sperm has a very streamlined shape that enables it to swim through the fluids of the vagina, the uterus, and then the oviduct, all the way here. It's a long way for the sperm to swim. The sperm's head, it has a haploid nucleus, and you already know now why it is the haploid nucleus. It undergoes meiosis, which is a reductive division. It undergoes meiosis 1 and meiosis 2 during its development. And by the time meiosis 2 has been completed, we have the half the number of chromosomes, and that's why it's called the haploid nucleus. And it has a tip here, which is called the acrosome. So this is the head of the sperm, and the head of the sperm has a nucleus where this haploid chromosome, haploid nuclear material is being carried, so 23 chromosomes here. And depending on whether the sperm has the Y chromosome or the X chromosome, eventually the child will become a male or a female. So it is the sperm, the genetic material of the sperm that defines whether the child will be a male or a female. So the nucleus carries the genetic material, and this is the head or the acrosome of the sperm. This the middle of the sperm contains mitochondria that use very high energy nutrients from the semen. So this is where the mitochondria are. Very large number of mitochondria are here. And then, when the sperm is formed, it travels via the epididymis, this long tube, and then it gets nourished by a number of fluids. And these fluids are generated, for example, by the prostate gland, as well as by the epididymal tissues themselves. And this liquid is called semen. So in the semen, there are a lot of nutrients which provide the energy that is needed by the sperm to travel. And the primary form of this nutrient is fructose. So these cells utilize fructose in order to generate their energy. And all this energy is basically coming with the function of the mitochondria that are found in the middle part of the of the sperm. And then we have a long tail, and this tail is a flagellum. It's called a flagellum. And this flagellum moves in a very nice coordinated manner, highly coordinated manner. And this movement is what requires a lot of energy. Movement of the flagellum is a highly energy intensive process. And this movement of the flagellum helps the sperm to swim the long distance that it needs to swim. There's one other very important point that we will make now, and that is, how does the sperm actually fuse with the head of with the egg, the surface of the egg. So when we look at the egg, obviously the egg is far, far bigger than the sperm, and you can see a little bit of that from this picture even. And one other important point to keep in mind is when fertilization happens, a single egg is released, obviously in most cases, and we will look at some exceptions later on. A single egg is released, and this single egg will be fertilized by a single sperm. Okay, so we don't have two sperms or more. If such an event happens, if two sperms end up fertilizing an egg, that particular zygote will be killed. It will not survive, okay, because the amount of genetic material will be more than what it should be, and therefore the embryo is not going, it doesn't even get to the stage of the embryo. Before that, it basically is degraded. So what we have in the egg is, of course, we have the genetic material inside the egg, we have the cytoplasm, and the egg has a jelly coat. So this coat needs to be breached. The sperm actually needs to pass through the jelly coat in order to release its nuclear material into the egg so that the fertilization can happen. So in the first stage, the sperm will squeeze through cells that are left over from the follicle. So the egg is actually surrounded by these follicular cells. And you know now what the meaning of the word follicle is. It is the structure in which the egg is matured in the ovaries of the female, and then it is released at the time of ovulation. And when the egg has been released, it is taken up by the oviduct. And at that stage, it is covered by some follicular cells. So you can see them labeled here, these gray cells. So the sperm has to pass through these cells that are left over from the follicle. And then the enzymes that we have in the head of the sperm, which you can see here, this is the acrosome. There are some enzymes that are found in the acrosomes, which can degrade the jelly coat. Once the jelly coat has been degraded, the two membranes of the sperm and the egg fuse. And once the fusion happens of the two membranes, the nuclear material, the genetic material of the sperm is released into the egg, and then the sperm and the egg chromosomes will mix together. So now we have 23 chromosomes coming from the sperm, 23 chromosomes coming from the egg, and we end up with the zygote nucleus containing 46 chromosomes. Okay, so this is what fertilization basically is. So the important point here is only a single sperm will fertilize a single egg, even though we have millions of sperms that are actually capable of fertilizing a single egg. Ultimately, only one will end up fertilizing it. So this is a picture of an egg that is surrounded by hundreds of sperm molecules, as you can see. This is something that happens in reality, but only one of the sperms will end up fertilizing the egg. The others will then be degraded and they will be lost. When we come to development, so let's say that the fertilization has happened. Now, what else is getting into the egg other than the nuclear material? Actually, nothing else. So the mitochondria that you have in the sperm remains in the structure of the sperm. It does not enter into the nucleus. And that is why, you probably already know this, that all the mitochondria that we have received, that we have in our cells, have come from our mother. Okay, so the mitochondria that were already found in the egg is then found in the zygote and in the embryo and then in the individual that is formed. So therefore, looking at the mitochondrial DNA, mitochondria has DNA in it, as you already know. Looking at the mitochondrial DNA gives us an idea of our maternal lineage, okay? Because none of, or very few of our mitochondria come from the father. Most of our mitochondria come from our mother. And that is why we can look at our maternal lineage, where we originate from, with the help of the mitochondrial DNA. So the key to development in all organisms is that it's very, very, very organized. So this is a remarkable process of signaling that happens, and a lot of it is still highly unknown. A lot of it is known in lower organisms. So for example, in zebrafish, in fish, or in flies, or in worms, we do know how the development happens in very good detail. But in humans, it is still highly unknown. With recent techniques such as single cell RNA sequencing, one can actually identify individual cells during development, obviously in model organisms. One can take individual cells in developing embryos and then determine the changes that are happening. So there's a lot that we have learned in the recent years, but again, a lot of it is in model organisms, and we still don't know much about how individual changes and how this level of organization is maintained during human development. The development begins with a cleavage, a series of very, very rapid cell divisions that results in a multicellular uh, ball of cells. And during cleavage, DNA replication, mitosis, and cytokinesis. So these are processes that you've already learned about. The replication of the DNA, cell division, and cytokinesis, where the two cells, mitosis, where we have the separation of the chromosomes after division, after replication, and then the cytokinesis, which actually leads to the cell division. It occurs very rapidly, but the total amount of cytoplasm remains unchanged. So if we look at the picture here, you can see that the size of the zygote does not change much. So this means that when the zygote goes from a single cell soon after it has been fertilized to 2 cells, to 4 cells, to 8 cells, and then to many, many cells, despite the division, we don't see the cell increasing in size, right? So this means that the cytoplasm does not increase too much. And this is a very, very unique way of dividing, whereby by the time we get to a blastocyst, the size of the zygote remains almost identical. Okay, so here this means that each cell division partitions the embryo into twice as many smaller cells. So the size of the cell decreases. And you can see that very clearly over here. You have 2 cells, then you have 4 cells, 8, and so on and so forth. But the size of the cell is much, much smaller in the blastocyst compared to the 2-cell stage or the 4-cell stage, etc. The cleavage continues as the embryo moves down the oviduct towards the uterus. So what is happening here is, so let me try to draw the oviduct again. So here we have the long oviduct, and then we have the uterus over here, and there is the other oviduct. So exactly which oviduct the fertilization will take place is random. So one of the ovaries releases an egg one month, the other ovary releases the egg the other month. So therefore, exactly which oviduct will have the fertilization is completely random. So when the sperm comes, the sperm will actually move to both the oviducts, and wherever it will find an egg, it will fertilize it. So let's imagine that it has fertilized an egg over here, and this egg has now started, zygote has now started cell division. So as this as this zygote starts moving in towards the uterus where actual implantation will occur, the cell division will continue. And then by the time it comes to the uterus, remember the size is the same because the cell division results in smaller cells, and the cleavage will continue as the embryo moves down the oviduct towards the uterus. So here we have the blastocyst, a hollow ball, which means that the inside we don't have any cells, but we have a sort of, if you can imagine a sphere, we have many, many cells on the surface of the sphere. If you can think of a tennis ball, inside is hollow, but we do have an inner mass of cells here. And the inner mass of cells are the totipotent stem cells. So these are the crucial cells that will give us the actual organism, the cells that are marked in purple over here. So this inner mass of cells will become the actual individual. The covering cells that are shown in yellow here will become the placenta, which will eventually nourish the fetus. So by the time the blastocyst actually comes to the uterus and then implants it, it looks somewhat like this. So quite a lot of cell divisions have already occurred. And by the way, one thing I forgot to mention, and this is something we learned last week as well, is this particular egg will finish its second stage of meiosis only after the fertilization happens. So keep that in mind. That is also something that is happening in the egg after fertilization. So fertilization has happened. The egg has completed its maturation. The zygote has been formed, and then the zygote has actually undergone the division. Before we move into implantation, I'll remind you how twins can happen, right? So maybe some of you have a twin in your own twin, or most of us know somebody who has a twin. So when we have fraternal twins, when we have identical twins, twins who are completely identical to each other, then at the two cell stage, these two cells separate from each other in a very, very chance event. This is not something that happens regularly, it's very rare. And these two cells will divide and they will develop independently. Okay, so they undergo a sort of restoration of the signaling processes, which will make them as individual cells, and then they will keep dividing. Okay, so these two cells are very, very similar to each other, right? Because they have formed from the same cell. So these cells now undergo mitosis over and over and over again in order to give us the blastocyst. So these two cells are pretty much identical to each other, which means when these two cells divide, separate from each other, I'm not talking about division here, they're separating from each other completely. And then they restart their whole cell division process and they become two identical individuals. That is how identical twins are formed. When it comes to non-identical, and one more thing, this also helps you understand why identical twins are always the same gender. You don't find one male and one female as identical twins. It's impossible. It's physiologically impossible. But you do find one male and one female twins who are not identical to each other. In that case, what has happened is the female has ovulated more than one egg. Okay, so this can happen either artificially. So when artificial insemination will happen, when in vitro fertilization occurs, then a woman can be given hormones which will make her ovulate more than one egg. This is possible. Or naturally, the female ovulates more than one egg. And if more than one egg is fertilized at the Sorry, was there a cut in the... Okay. Okay. Okay, so for non-identical twins, what is happening is two different, let's come back here, two different eggs are fertilized by two different sperms. And so the process of formation of zygote 2, 4, 8 cells stages, they all happen for two embryos that have been fertilized, or actually more. It doesn't have to be twin, it can be triplets, it can be quadruplets, it can be many, many. Okay, Hande. It can be many different, it can be many eggs that can be fertilized. So as many as 3 or 4 or even 5 fertilizations have been reported occurring naturally in the news. However, it's very, very, very rare. Triplets is extremely rare. Twins are relatively more commonly seen. We will talk about in vitro fertilization towards the end of the lecture. So when we have the blastocyst, this is, remember I mentioned that these are the cells that will form the actual individual, that will form the organism. These cells are totipotent stem cells, which means that these cells will divide into any type of cell in our body. So the initial differentiation that happens results in the formation of 3 layers of cells, and you can see them here in blue. we have the ectoderm. In pink, in the middle, we have the mesoderm. And in yellow over here, we have the endoderm. So the ectoderm will form, as the name perhaps suggests, ecto means outside. Ectoderm will result in the formation of the skin. The endoderm will result in the formation of organs such as the inner lining of the intestinal tract and the liver. And the mesoderm will end up forming our skeletal system, the muscle system. Okay? So each of these individual cells will keep dividing and they differentiate into the different organs that we have during development. So this also suggests that there is a very, very well coordinated, very well organized, and extremely regulated process that is happening from the single cell stage to the blastocyst stage, and then eventually for the development of the actual individual. A lot of things can go wrong in between. So by the time the implantation happens, many of these embryos are actually lost. So if there are any genetic problems, if the development until that stage has not happened, then the embryo may be lost. And as many as 1/3 of the embryos that are fertilized are actually lost before they can be formed, before they can be, they can develop into an organism. This means that very often women don't even know that they are pregnant because this implantation happens about 8 to 10 days after fertilization. And when the embryo is lost, it is lost through, because as soon as the embryo is lost, the levels of estrogen and progesterone fall, which will lead to the initiation of the menstruation, and therefore this embryo is lost via the menstrual blood. So often women don't even realize that they were pregnant, and they just think that they had their menstruation and the embryo is lost. Often, if the woman is aware that she is pregnant and then she has a menstruation, then this is called an abortion. And if it occurs spontaneously, a spontaneous abortion can happen as late as 6 weeks after fertilization, even, or 10 weeks after fertilization. In this case, really, it's very likely that there are gross genetic disorders in the embryo, and therefore it is not viable and it was unable to survive. So about 6 to 7 days after fertilization, the embryo has reached the uterus. Okay, so remember, it travels through the oviduct all the way to the uterus, and it's now a fluid-filled hollow ball of about 100 cells called a blastocyst. So here we are. So this inside is hollow, but it is filled with the fluid. It's not filled with cells. And then we have the inner cell mass, and then we have the outer layer of cells. So this then gets implanted, and so the inner cell mass will eventually form the fetus. So here let's have a look at this picture. This is embryonic development in the sea urchin. So this is not human, obviously. This is an invertebrate. Let's see how it happens. This is the first division. And then we have the second division, and you can see very nicely how, and then you have the inner cell mass that you can see over here. Where did it go? It was there. Okay, so here, there you go. You can see the inner cell mass beautifully. Such a lovely picture. Look at that. Okay, so this is how the initial development happens. And here we have a frog developmental picture. Very, very rapid cell divisions happening here as well. So as I was mentioning, not all embryos are capable of completing the development. If the implantation of the embryo occurs in the oviduct, so I've already drawn how the oviduct looks, then this is called an ectopic pregnancy. Ectopic means that the embryo has implanted itself in a location different from the uterus. This is very fatal to the embryo if the implantation happens in the oviduct, because obviously the embryo cannot develop in the oviduct, so the embryo invariably dies. And it also is very, very dangerous for the mother, and it requires immediate medical attention so that the embryo can be removed, the dead embryo can be removed from the oviduct of the female. Many other embryos, as I was mentioning, they spontaneously stop developing, either because of chromosomal abnormalities or because of developmental abnormalities. So as they divide and divide, some abnormalities come up. So a spontaneous abortion or miscarriage occurs in as many as 1/3 of all pregnancies. And often this happens before the woman is aware that she is pregnant, because of the way in which the menstrual cycle happens. Remember, ovulation happens soon after the menstruation is completed. And then by the time the woman has her second menstruation, the next menstruation rather, there's a period of about 28 days in between. So by that time, if the fertilization has happened and the blastocyst stage has been reached after 7 or 8 days, and then it fails to implant, then it will appear as a normal menstruation for the female, and she won't even be aware that she was pregnant. So about 9 days after conception, the cells, so conception is of course the fertilization, the cells of the early embryo begin an organized migration. So this organized migration is something that we saw in those two videos, in actually the first video, the sea urchin video, where this organized migration leads to the formation of gastrula, giving the three layers that we talked about: the ectoderm, the endoderm, and the mesoderm. So the ectoderm forms the skin, and it develops also into the nervous system. The endoderm forms the inner lining of the digestive system and the organs such as the liver, also pancreas, thyroid. And the mesoderm will give us the heart, the kidneys, the muscles, and other organs and tissues. Most of the other organs and tissues develop from the mesoderm. So now many cellular changes are seen in the embryonic structure, and all of this is highly regulated, extremely well coordinated biochemical signals that pass between neighboring cells. So there are actually cell-cell synapses. These can also be called synapses because the cells come very close to each other. There's signaling between neighboring cells, whereby these embryonic cells, these are totipotent embryonic cells, they can be informed precisely what to carry out. So which signals will turn on? Because remember, genetically every single cell is identical. So how is it that the mesoderm cells will give us the muscles, but the ectodermal cells will give us the neuronal cells? So each cell has to be activated via a very specific set of signals, which will make it differentiate into a specific type of cell or tissue. So there are other signals also that can cause the cell to change its shape or to migrate from one location to the other. So as the fetus develops, cells need to move to their correct location, right? Your liver has to be in the location of the liver, your brain has to be in the location of the brain. So therefore, it's very important that these cells move in a coordinated manner, and the extracellular matrix that we have helps these cells to actually move to their correct location. Because if they don't move to the correct location, the organ will not be developed at the correct location, right? So you don't want your liver in your brain. Your liver has to be where the liver is, your brain has to be where the brain should be. So these movements also will result in the correct location, in the correct, in the place where the cell should be. Another key developmental process is cell death. So although it seems counterintuitive, right? How can you develop if the cells are dying? But this is what regulation is. So some genes encode proteins that will kill certain cells in order to give us our hands and feet so that the fingers and toes can be separated. So let's look at a picture. If we do not have apoptosis, if we do not have cell death, then we do not develop fingers, right? So it looks like a stump here. But with regulated, programmed cell death, we can actually have these individual fingers and toes in our hands and feet by the process of cell death. So this killing of selected cells is also highly regulated, because obviously you don't want, only those cells that are meant to die should die, others should not. So pregnancy or gestation is carrying the developing young within the female reproductive tract. Let's take a quick break. We're all actually already out of time for the first hour. So I'm going to stop here. And you can turn off your cameras. It's fine. You don't need to keep your cameras on. And we will see, it's 9:32. So in about 10 minutes, I'll see you guys again.